1812 treatment on plasma biomarkers in lower p-tau 217 subgroup aligns with clinical benefits in mild to moderate AD patients.
Thank you. It's a pleasure to be here. Tell you about our SHINE trial testing the effects of zervimesine in patients with Alzheimer's disease over a 6-month course. Here are my disclosures. So zervimesine, or CT1812, is an investigational therapeutic. It's an oral, once-a-day, small molecule sigma-2 receptor modulator, and it binds to the sigma-2 complex shown here in purple, which leads to a modulation which kicks off, if you will, the A-beta oligomers from the neuronal synapses. And so there's a host now of preclinical and clinical evidence that we've published that shows this and demonstrates this nicely, both clinical data as well as preclinical. So the mechanism of action is a synaptoprotective action to slow neuronal injury or loss. And I want to point out that our mechanism of action is distinct from anti-amyloid immunotherapies. So here's our trial design. As I mentioned, it's a 6-month course. There is a randomization of patients into placebo arm or testing 2 doses of zervimesine, 100 milligram once a day or 300 milligram. 153 participants were randomized across 30-plus sites globally. This is important that all patients in the trial were mild to moderate with MMSE score ranging from 18 to 26, with an A-beta positive biomarker criterion for inclusion, either as assessed by amyloid PET or a CSF amyloid test. And the assessments, this study seeked to assess safety as well as signals of efficacy using various measures. The ADAS-Cog11 was the primary efficacy outcome measure. We also looked at the mini mental state exam, MMSE, activities of daily living, and the clinical global impression scale. And in this analysis, some of this data has been shown at prior meetings, the intent to treat group and the phospho-tau subgroup analysis of the clinical data, but today I'll take you through that and focusing in on the biomarker, plasma biomarker results from this subgroup analysis, which is quite exciting to us. So in brief, the safety and tolerability have been previously described and shown at AAIC and CTAD, but as you see here, there's a favorable safety profile that was exhibited by zervimesine across this trial. No discontinuations due to adverse events were found in the 100 milligram dose. Some discontinuations were in the 300 milligram dose, and as you can see, there's approximately equal percentage across placebo and zervimesine treated patients, shown here. So this session has nicely, I think, introduced the phospho-tau 217, the plasma measurement that can be done. We, in this trial, used the ALZpath Simoa assay in collaboration with Charlotte Tennyson's group. And just to recap what we've already learned today, plasma phospho-tau 217 can be an indicator of the degree of amyloid pathology in the brain, and we can detect that indirectly with the blood test, so that's quite nice. And prior data with anti-amyloid lowering therapies have demonstrated that there's precedence for the fact that with patients with lesser pathology, lower tau, for example, in the Trailblazer 2 donanemab trial, they demonstrated that there was a larger degree of slowing in the iADRS in the low tau group compared to the high tau group. And given our mechanism of action in binding sigma-2, which then displaces amyloid beta oligomers from synapses, which we've seen can lead to increased levels in the CSF, as previously published, we hypothesized that in this trial, patients treated with zervimesine might exhibit a greater cognitive benefit. So the baseline characteristics of the trial are shown here. You have the intent to treat group on the left column, modified intent to treat, the below median phospho-tau 217 and the above median phospho-tau 217. And the median for this trial, for the subgroup analysis, which was pre-specified, ended up being 1 picogram per mil. So that's the cutoff using this assay. And you can see here that the percent of patients that are female are approximately similar. The ApoE status is approximately similar across these 3 groups, cohorts. So to take you through the cognitive data briefly, you see here that there was a tendency for a favorable improvement. If you look on the left graph in the modified intent to treat population with 38% slowing, but this was not significant. But you can see the gray line is the placebo trajectory, where there's cognitive worsening over 6 months, and there's, again, the 38% slowing in patients treated with zervimesine, although not significant. But what we noticed was in the low tau group in the center panel, this favorable tendency became larger. And if you look at the levels in the zervimesine group in the middle graph here, you see that there's really a preservation. There's no decline, and if you look at the orange line here, there's no decline at 6 months compared to the patient's baseline in that treatment group, as compared to the placebo, which steadily declined over the 6 months. And as you see on the right with the high tau group, there was really nothing remarkable to speak of. The placebo and zervimesine overlapped. We noticed a similar trend when we looked at the mini mental state exam results. You'll see the intent to treat data on the left and the low tau group on the right. And you can see in the intent to treat, it's approaching a trend of a 70% slowing. And then in the low tau, the effect size again became larger, again approaching a trend of 108% slowing. And then here, what you'll see in the activities of daily living on the left, and this is focusing now on the low tau group only, this is what's shown on this slide, you see that activities of daily living, there seemed to be a favorable directional effect, as well as in the clinical global impression scale on the right-hand side, with the trend observed at 6 months. Now, turning to the biomarker results, we assessed plasma biomarkers in these patients and the intent to treat group, and that's shown in the table on the far left column. See it, modified intent to treat. The low tau group is in the middle, and the above median or high tau group is on the right. And what you see here is that largely there was weren't too many remarkable findings in the intent to treat population. Other than the brain-derived tau, which we assessed, which is a trend, a p-value of 0.09. But in the low tau group, what you'll notice is with the A-beta 42, 40, GFAP, and NFL, the effect sizes are all, and phospho-tau 217. If you look at the effect size, which is on the top line of each of these in the table for each of the cells, you'll see that the effect size is greater in the low tau group. And you'll see what's highlighted there in orange are trends that we were able to see that we weren't able to see in the intent to treat population. And notably, GFAP, a neuroinflammation marker, you see is now significant in this case. And if you look at the high tau group, there was really nothing, again, that was remarkable. So this was quite interesting, and just to take you through the plots very briefly, here you can see the effect that zervimesine has on the amyloid beta 42 and 40. 40 is on the top, 42 in the bottom. You'll see the placebo in gray over the 6 months, this change from baseline, and you'll see the treatment effect group in orange. And so focusing on the GFAP, you'll see here that in the intent to treat group on the left, there's really nothing too remarkable about a change with a treatment effect with GFAP, but in the low tau group on the right, this now again becomes significant with a significant change compared to placebo. Looking at the NFL, you'll see here that there's a directionally favorable effect on NFL, the marker of neurodegeneration in the intent to treat population on the left, but on the right you'll see that this effect size grew and a trend was observed, a favorable trend. And lastly, we looked at the phospho-tau 217 levels, and you see here that, again, the effect size in the low tau group on the right is larger and approaching a trend in this case. So overall, I think what's remarkable about this study is that across these measures of cognitive and functional outcome measures, as well as the biological impact as assessed by plasma biomarkers, is consistently favorable with greater effect sizes seen in the low tau cohort. So baseline plasma phospho-tau enabled us to identify this subgroup analysis, to identify a strong responder group of patients that might be more likely to respond to zervimesine, which is now the hypothesis and what we're planning for phase 3, at least under discussion, of whether or not we stratify using phospho-tau 217 or do we enroll based off a set threshold of lower than, say, 1 picogram per mil phospho-tau 217. Those kind of discussions are ongoing. So in conclusion, zervimesine was found to be safe and tolerated in the SHINE trial. Robust cognitive and functional impact was observed in the signal-seeking trial in the pre-specified below median plasma phospho-tau 217 subgroup, with the prominent effects on neuroinflammation with GFAP, neurodegeneration with NFL, and also amyloid biology, consistent with a favorable clinical outcome. So thank you. Open to questions.
Thank you, Mary. We have time for a couple of questions.
Thank you for some very encouraging results. In the other studies that have looked at tau, they've been using tau PET to discriminate between high and low, and you're using phospho-tau 217 that some have suggested is really more indicative of amyloid positivity than tau positivity. Do you have any data on tau positivity from CSF samples or tau PET that would confirm that what you're looking at is actually a change in tau rather than just low A beta to begin with?
That's a great question. We have not looked at that yet. I think we do have some measurements that we can now go back post hoc and look at baseline tau levels in the trial, but we didn't do a PET tau PET imaging in this study. We won't be able to directly relate it back. I think some of the studies that, though, the monoclonal antibodies did assess, again, I think after, I don't know if it was pre-specified, but looked at first tau PET, but then later looked at the plasma phospho-tau 217. And I believe in the lower cohort, the lower level subgroup, that the increased benefit was still observed.
All right, thank you.